Proteins act as enzyme catalysts that increase the rate of most chemical reactions by lowering the activation energy without being changed by the reaction. In this lab, we used the enzyme trypsin, which helps to digest proteins by breaking down food into small enough molecules for absorption into the body. Trypsin is active in the small intestine at a pH level of about 7.5 and in the pancreas at a pH level of about 7 in animals. Bacteria secrete trypsin for use in other locations. In this lab, we asked the question, how do varying temperatures affect trypsin activity? We hypothesized that as the environmental temperature increases, the effectiveness of trypsin in digestion increases until a certain temperature when the enzyme denatures and becomes ineffective. We know that protein denaturation occurs because of weak bonds and an increase in random molecular motion due to the temperature conditions. We tested this hypothesis by using the temperatures 27, 37, 47 degrees Celsius in the first experiment and 47, 57, 67, and 77 degrees Celsius in the second experiment. First, we performed one negative control for every temperature. The negative control includes 450 microliters of buffer of pH 8 and 0.15 NaCl, and 500 microliters of azocasein with 50 microliters of water instead of trypsin. Then we filled 2 centrifuge containers for each temperature in the first experiment and 3 centrifuge tubes for each temperature in the second experiment. We filled them with 450 microliters of buffer, 500 microliters of azocasein, and 50 microliters of trypsin. After all of the tubes were incubated for 20 minutes, we added TCA to stop the reaction. The tubes were centrifuged for 2 minutes at 12,000 RPM rates per minute to separate the pellet, which is the undigested substrate, from the supernatant. We added each supernatant to their individual cuvettes and blanked the spectrophotometer at 450 nanometers with water. The azocasein allows the test to absorb at 440 nanometers. Lastly, we read the absorbance of each supernatant and recorded the data. According to the data, the average absorption is the highest at 47. At 47, the average is 0.577, and from there it decreases, although the standard deviation is also highest. So we had high variability between each test absorption within the 47 degrees Celsius temperature. The p-values that were calculated showed that there was no statistical significance between any of the temperatures besides 47 and 77, so we cannot conclude that 47 is our optimal temperature. But we can still say that the average absorbance was highest at 47 degrees Celsius and decreased from there, which slightly supports our hypothesis. So we can see from this graph that it is overlapping between 47, 57, and 67, but where it's at 77, the p-value is not overlapping 47. And we have a data chart of all of our p-values that we found. And we had a few problems along the way of our experiment. First being that we used 2 tests and 1 negative control for each temperature in the first experiment, which did not give us a clear average for absorbance. So we decided to use 3 tests and 1 negative control for each temperature in the second experiment. Another problem that we had was we accidentally pipetted 500 microliters of buffer instead of 450, so we wound up taking out the extra 50 microliters. And lastly, the water bath that we set for 57 degrees Celsius decreased to about 44 degrees Celsius, so the measurement for that temperature had to be redone.